All right. Today, we are stepping right into the lab to talk about a pretty fundamental challenge in science. We're surrounded by this entire universe of life that is completely invisible. So how on earth do we even begin to study it?
And that's the real question, isn't it? To understand bacteria, how they work, what they do, how they affect us, you first have to get them to grow somewhere you can actually see them. And that's exactly where the art and, well, the science of bacterial culture comes in.
Okay, so first things first, we need to set the stage. Bacteria are, and I mean this literally, everywhere. So if we want to study just one specific type, we can't just leave a petri dish out on the counter. We have to create this perfectly controlled, sterile environment, a total blank slate really, ready for our microscopic star to take the spotlight.
Now, the first big decision a microbiologist has to make is where the bacteria are going to live. Are we going to grow them on a solid surface, like the agar in a petri dish? Or should they be floating around in a liquid broth inside a flask? The little sample of bacteria that we start with is called the inoculant, and whether we choose a solid or a liquid, well, that all depends on what we're trying to find out.
So we've got our sterile stage, but now we have to cook dinner. Seriously, growing bacteria is a lot like being a microscopic chef. You have to prepare the absolute perfect, custom-tailored menu for your tiny and sometimes very picky guests.
And this leads to two main styles of cooking, if you will. On one hand, you've got defined media. Think of this like a super precise high-end recipe, where every single chemical, from the glucose to the salts, is known and measured down to the microgram. Then you have complex media, which is, well, it's more like a rich, all-you-can-eat buffet. It's got stuff like yeast extract or tryptone, which is basically digested milk protein. We know it's packed with nutrients, but the exact chemical breakdown, it's a bit of a mystery.
So you might be wondering, why even bother with the messy buffet when you can be so precise? Well, it turns out some bacteria are what we call auxotrophs. Just think of them as incredibly picky eaters. A lot of times, these are pathogens that have spent so long living the easy life inside a host that they've actually forgotten how to make their own essential nutrients, like certain amino acids. For them, that complex, nutrient-packed buffet is the only meal they'll even touch.
And this is where it gets really clever. Scientists can actually design these menus to be powerful tools. A selective medium, for example, acts kind of like a bouncer at a club. It only lets certain types of microbes in. If you make a medium where methanol is the only food available, guess what? Only methanol-eating bacteria are going to grow. Then you've got diagnostic media, which is like a built-in litmus test. For instance, the bacteria Staphylococcus aureus will actually turn a specific medium a distinct gray-black color, giving you a visual confirmation right away. How cool is that?
All right, the meal is cooked, the table is set, now it's time to seat our very special guest of honor. And this is the most delicate step of all. We have to transfer the bacteria we want to grow onto their new food, all without letting any of those unwanted germs floating in the air crash the party.
The name for this whole procedure is aseptic technique, and it is, without a doubt, the absolute bedrock of microbiology. It's all about maintaining purity. You can really think of it like performing surgery. Every tool has to be sterile, and every single move is precise and deliberate to keep any contamination out of our pure culture.
And the process itself, it's like a careful, methodical dance. You start by sterilizing your tools, usually with a hot flame. Then you use that perfectly sterile tool to pick up a tiny, tiny sample of your bacteria. Next, you gently transfer that sample to the new medium, and then you seal the whole thing up, fast. Every single step is a defense against contamination, all to make sure our culture stays pure.
So, success! Our bacteria have grown, we've studied them, now what? We want to save our hard work, right? But you can't just pop a petri dish in the fridge and call it a day. We need a way to put these organisms into a state of suspended animation, basically creating a living library of microbes for future study.
The secret to long-term storage is pretty simple in concept: extreme cold. The whole goal is to just stop all biological processes so the cells don't age or die or mutate, but you have to do it in a way that doesn't actually kill them. We're trying to pause life, not end it.
And when we say extreme cold, we really mean it. A standard freezer in a microbiology lab is set to minus 70 degrees Celsius. And to stop ice crystals from basically shredding the cells as they freeze, they're suspended in a 50% glycerol solution, which acts like a kind of biological antifreeze.
Now, the method you pick really depends on your timeline. For short-term stuff, just a few days, you can just transfer some bacteria to a fresh plate. For long-term storage, like years, that minus 70-degree freezer is the go-to.
But for the absolute ultimate in preservation, scientists use lyophilization, or freeze-drying. It removes all the water and can store a culture almost forever. It's like a microbial time capsule.
Of course, waking them up is just as delicate. When the cells come out of that deep freeze, they are in a starved, super stressed state. You can't just shock them back to life with a bare bones, minimal medium. No, they need a gentle re-entry, a rich, complex welcome back meal to give them the best shot at recovery.
This brings us to the final, and you could argue the most important part of all lab work, sterilization. It's the bookend for everything we do in the lab. We sterilize our media before we start to guarantee a pure culture, and we sterilize all our waste after we're done to make sure everything is safe.
The absolute gold standard for sterilization is a machine called an autoclave. It's basically a giant, high-pressure steam cooker. It gets up to 121 degrees Celsius for at least 15 minutes. That combination of intense heat and crushing pressure is enough to kill everything. All living cells, all viruses, and even the toughest life forms on the planet, bacterial endospores.
But what if your stuff can't take the heat? I mean, some things just melt. Well, for heat-sensitive media, you can use something called tyndallization, which is a clever cycle of heating and cooling that tricks spores into waking up before killing them. Radiation, like UV or gamma rays, is great for zapping surfaces or prepackaged items. And for really delicate liquids like vitamins, you can just filter them through a superfine sieve that physically pulls the microbes out instead of killing them.